Chemolithoautotrophs are microorganisms that obtain carbon from inorganic sources using chemical energy, such as oxidizing iron, hydrogen sulfide, and ammonia. This allows them to live in harsh environments such as subterranean and deep-sea ecosystems that function in the absence of sunlight or with oligotrophic conditions. Studying these organisms can provide insight on how alternative non-photosynthetic primary production and metabolic strategies can support survival. Furthermore, the presence of chemolithoautotrophic microbial communities in these extreme environments develops an understanding of how higher trophic levels can be supported in ecosystems such as subsurface soils, dry deserts, and possibly even extraterrestrial environments.
Deep-sea hydrothermal vents are areas that have an influx of fluid that is flowing up through cracks where magma is running underneath the ocean floor. This fluid is highly anoxic, acidic, and high in concentrations of minerals such as hydrogen sulfide, methane, iron, zinc, and copper. As the hot water with the dissolved minerals meets the cool ocean water, a huge cloud of precipitate forms, which has been nicknamed the black smoker if it has iron. The white ones can be formed with large amounts of calcium or silica. In this cloud, the reduced minerals from the fluid serve as electron donors, and oxidized seawater serves as electron acceptors. This combination creates the foundation for a community of chemoautotrophs. These communities are highly diverse, though the exact content will change based on geographic region and magma source. These chemoautotrophs then serve as the primary producer for the ecological community, in which heterotrophs can gain energy through methods such as direct consumption, microbial gardening, or symbiosis. Invertebrates will use microbial gardening by consuming the microbes but leaving a small amount to grow the next crop. One invertebrate, known as the tube worm, uses symbiosis by having the inside of the organism colonized by these chemoautotrophs, which will then produce organic compounds to feed the worm.
Acid mining drainage, or AMD, is a serious environmental problem that results from mining activities. While some cases of acid mining drainage are natural, most are due to human activities. Mining exposes sulfide minerals such as chalcopyrite, chalcocite, spherulite, and galena to air and water. The exposure leads to biologically mediated acid generation, which produces acidic leachates high in toxic metals. Microorganisms populate the AMD environment and are responsible for driving community development. To better understand the ecological and evolutionary process behind this, researchers have used metagenomic analysis. In one study, researchers obtained 76.2 million base pairs of usable DNA sequence from an AMD environment. They reconstructed these sequences to give nearly complete genomes for 2 iron-oxidizing bacteria, Leptospirillum group 2 and Ferroplasma type 2, as well as partial recovery of 3 other genomes. Fluorescent in situ hybridization analysis confirmed the presence of these organisms, with the Leptospirillum group 2 being dominant at 75% of the community, while the Ferroplasma type 2 represented approximately 10%. Interestingly, the Leptospirillum group 2 has a lot of CO2 fixation pathways, making it chemoautotrophic. On the other hand, the Ferroplasma type 2 has the mechanism to fix CO2, but contains sugar and amino acid transporters, making it a heterotroph. Neither of these bacteria have N2 fixing genes, but Leptospirillum group 3 has some. This means it could be a type of keystone species in the AMD community. The metagenomic analysis also revealed that the biofilm in the AMD environment had low diversity and low amounts of gene rearrangements, which made the analysis successful. Overall, metagenomic analysis is a powerful tool for understanding complex microbial communities and their interactions in the environment.
Chemolithoautotroph colonies can also occur in carbonate caves formed within karst terrain, which is the result of the dissolution of bedrock in water. These cave systems are subterranean and aphotic, meaning they're unable to support photosynthetic primary production. In many caves, organic carbon will come dissolved in water that drips from the surface, forming speleothems, which are similar to stalactites. However, in desert carbonate caves, the surface water that carries organic carbon is limited, creating an oligotrophic environment below. As a result, a limited number of heterotrophic colonies can survive on speleothems. However, many of these colonies are entirely autotrophic, and evidence suggests that they obtain their energy via nitrate and iron oxidation. Interestingly, after metagenomic analysis of the community on speleothems, specifically in the Kartchner Caverns in Arizona, it was revealed that 10% of the genomes sampled from speleothems contained archaeal DNA, which is a significant increase from the average 2% archaea abundance in soil. It is expected that these archaea are the primary producers of the ecosystem through ammonia oxidation, as half the analyzed ammonia oxidation genes were classified as archaeal. Additionally, more in-depth analysis detected genes for all 6 known carbon dioxide fixation pathways and suggested that the community genetically adapted to the low nutrient conditions. This is a similar ecology to microbial slimes located in subterranean aquatic environments, in which production is driven by inorganic nitrogen cycling through ammonia oxidizing archaea, illustrating that this type of community can exist in both terrestrial and aquatic ecosystems.